Hello everyone, my name is Adnab Oluchi Colette, and today my presentation is about the expression of human epidermal growth factor 2 receptor gene, which is also known as the HER2 gene in mammalian cell. The objective of this project is to investigate the role of HER2 gene in cancer progression, as well as its potential as a therapeutic target. Our gene of interest is the HER2 gene, and it's also known as the ERBB2 gene. It is a growth factor receptor gene that produces HER2 proteins. Now, it encodes a receptor tyrosine kinase that plays a critical role in regulating cell growth and differentiation. Overexpression of this HER2 gene is often associated with aggressive breast cancer and other types of malignancies. The sequence displayed here is the complementary DNA strand of this HER2 gene, and it is used in this experiment for cloning and expression in the mammalian cell. Primer design and ligation. To successfully clone HER2 gene, forward and reverse primers were carefully designed. The forward primer included a COR1 restriction site, the COSA sequence for efficient translation, and the start codon, which is the ATG. The reverse primer contained the BAMH restriction site, the stop codon, as well as a purification histag sequence for downstream protein purification. This setup ensures proper orientation of the gene into the multiple cloning sites of the vector and its efficient expression. Place of expression plasmid. The pcDNA 3.1 plasmid was selected for this experiment due to its high level expression capacity in mammalian cells, driven by the CMV promoter. Not just the CMV promoter, it had an additional T7 promoter. Also, the plasmid contains multiple cloning sites for easy gene insertion and antibiotic-resistant marker for selection of transformed cell. Examples of antibiotic selection marker in this plasmid were the ampicillin and the neomycin. The cloning strategy employed involved the PCR amplification of HER2 gene, the restriction digestion, fragment purification, ligation into the pcDNA 3.1, and as well as the bacterial transformation. Also, the plasmid were extracted at the end. Here is a simplified map of our recombinant plasmid. Our plasmid of choice was pcDNA 3.1. Now, within the multiple cloning side, we had our restriction site, which is the ACOR1 and the BAMH1. The HER2 gene was inserted between the ACOR1 and the BAMH1 restriction sites within the multiple cloning site. The histag was also added before. The histag was also added before the EGFP protein, which is the enhanced green fluorescence protein. Now, this placement allows for the purification of our fusion protein, which is with the aid of the histag, as well as the fluorescence functionality and visualization of our protein using with the aid of the EGFP protein. Also, the choice of ACOR1 and BAMH1 as a registration site enhanced efficient cloning without disrupting the essential regulatory elements. Transformation, colony screening and verification. To transform competent E. coli, we follow the standard protocol, which involved the preparation of our competent E. coli cells, then heat shock, recovery, and plating on the LB ampicillin plate. To verify successful transformation and correct ligation, we isolated the plasmid DNA, digested the ACOR1 and the BAMH1 restriction enzyme, and run on Agarose jet to confirm the fragment sizes. Finally, the Sanger sequence was conducted using a primer in the CMV promoter region to verify the integrity of the HER2 gene inserts. Functional expression and validation. To validate the functional expression of HER2 protein, a Western blot analysis was performed using antibodies targeting the HER2 gene, this tag, and the EGFP protein. Also, a tyrosine kinase phosphorylation assay was also performed to assess the HER2 activity. Fluorescence microscopy was also employed to observe the GFP fluorescence, confirming the successful protein expression and localization. In conclusion, through this project, we demonstrated a robust approach to clone and express HER2 gene in mammalian cells. This result contributes valuable insight into the HER2 functional behavior and its visualization using EGFP. paving way for further cancer research and therapeutic development. Thank you.